Hello friends, welcome back to my channel. In this lecture, I'll be talking about the points of difference, different types of polymerase and replication factors that are used in prokaryotes and eukaryotes. So, to start with, let's talk about helicases. Helicases are the ones that will separate the double stranded DNA, and you will get two single strands of the DNA based on which the complementary strand can be synthesized using these two templates, right? So, in prokaryotes, that job is done by helicase DNA B, while in eukaryotes, we have mini chromosome maintenance protein 2 to 7. Okay, it's helicase in eukaryotes. So, talking about single strand DNA binding protein, okay, this single stranded DNA, okay, has to be held in place rightly. That is done by single strand binding proteins in prokaryotes while replication protein A in eukaryotes. The primase activity is shown by DNA G in prokaryotes like E. coli, and Pol alpha itself shows primase activity as well, because that's the one that will add the RNA initiator. Later, it also shows polymerase activity wherein it adds initiator DNA, and later the polymerase, actual polymerase, will take over, right? So we had actually spoken about, we had discussed this in previous lecture videos. You guys can go and check that out. Sliding clamp in prokaryotes is beta clamp, while in eukaryotes we have proliferating cell nucleoantigen, that is PCNA, while the clamp is loaded in prokaryotes by gamma complex, okay, gamma delta complex, and replication factor C is required by PCNA so that it is loaded. So loading clamp in eukaryotes is replication factor C. Later, the polymerase, actual polymerase activity is shown by Pol 3 in the case of prokaryotes, while Pol delta, it's recently seen that Pol delta itself replicates the leading and lagging strand. Although it was previously thought that Paul epsilon used to synthesize the leading strand while Paul delta was involved in lagging strand synthesis, it is now seen that both the jobs are done by Paul delta itself. Now, proofreading activity is done by epsilon Pol 3 subunit, okay, while the part of delta subunit has proofreading activity, which is nothing else but 3 to 5 exonuclease activity, okay, of this delta subunit. So proofreading in eukaryotes is delta subunit. Who removes the primers? Okay, in the case of prokaryotes, we have Pol 1, and it can be sometimes also the RNA H, okay, that removes the primers, while in eukaryotes, okay, the flab endonuclease 1 removes the primers. Gap filling is done by Pol 1 in prokaryotes, while it's done by Pol delta solely in eukaryotes. And ligation is done by DNA ligases, wherein the last phosphodiester linkage is made and it is ligated. So hope this video summarized the whole procedure of eukaryotes and different types of factors that we use, because this is the very important part wherein you get it on just one slide. You guys can take a screenshot and write it down again, okay? It will be very helpful during your examination. Thank you.